Professor Ray Stallings is a professor of cancer genetics on RCSI and is the current director of research at the college. He's an internationally recognized leader in the area of cancer genomics, and has had leaderships in institutions across the world, including a period in the University of Texas Health Center at Houston. So his most recent research of functional high throughput screening reveals miRNA and for neuroblastoma, and it epitomizes the modern translational approach to finding solutions to solve current health issues. And we hope this talk will inspire you. So I'd like to welcome Professor Stallings, and if there's any questions, we'll answer them at the end. Thank you.
Thank you very much for that introduction. And also, I'd like to thank the organizers of the meeting for the invitation to talk today. This has been a really well-organized meeting, and it's very much appreciated. So I think I will just open up my slides first. And there we go. Can everybody see that now?
Yeah, I can see them perfectly.
Okay, I assume so. So just to give you a little bit of idea of what I'm going to talk about, I was asked to talk a little bit first about my career paths and choices that I've had during my career. So I'll do that a little bit, and I might keep referring back to it during the talk. The main part of the talk is on the childhood cancer called neuroblastoma, which is a heterogeneous disease. And because there's a fairly large number of undergraduate and postgraduate students in the audience, throughout the talk, I'll probably pepper it with a little bit of career advice, things that I have found useful during the development and progression of my career. So just to recap my career path, I've actually had 3 different careers in science, and I'll tell you a little bit about that. I started my professional career as a research scientist at the Los Alamos National Laboratory in New Mexico. Even though I'm not a nuclear physicist, I did work in their biology division. And when I was there, I worked on basic research on the Human Genome Project. This was an early stage of the project to map and sequence the human genome. I was also a professor at the University of Pittsburgh Medical Center, also doing genomics research. And then I shifted my career from being a basic biomedical research scientist to being the chief cytogeneticist at Our Lady's Hospital for Sick Children in Dublin. That was a major career shift from basic research to being a clinical scientist and being in charge of a very large diagnostic cytogenetics lab that provided services for the entirety of the country of Ireland. I then moved back to being a translational researcher in medicine as a professor at the University of Texas Health Science Center at San Antonio. And then Ireland wanted me back, so I moved back to Ireland. So in total, I've lived in Ireland for 23 years, and I became a professor of cancer genetics at the Royal College of Surgeons in Ireland. And then I had my 3rd career change, which was to become the Director of Research and Innovation at RCSI, where I was a very high-level university official in charge of research, and I was in charge of a research institute that had more than 500 active research staff. During that time, I also carried on with my neuroblastoma research. So just to reiterate, as I said, I did start out studying the human genome at Los Alamos and at the University of Pittsburgh. We developed a technique, in fact, to do physical ordered clone mapping of the human genome, which we published in PNAS. And I was actually quite proud to be part of this massive endeavor to sequence the human genome, because sequencing the human genome has provided an unparalleled opportunity for studying the genetics of human disease, phenotypic diversity, and evolution. All of this was eventually published in Nature, and I am one of about 4,000 authors on this Nature paper. But it was probably one of the most important projects in translational medicine and basic research in the last 100 years. So when I moved to Ireland, and just as a personal matter, my wife is Irish, so that was part of the attraction to moving here. Although I absolutely love Ireland. I became director of clinical cytogenetics in Our Lady's Children's Hospital in Crumlin, and I was the inaugural director of this laboratory once the National Center for Medical Genetics was formed. But I became introduced to a tumor called neuroblastoma, which arises from primitive cells of the sympathetic nervous system, and it accounts for 15% of childhood cancer deaths. Now, neuroblastoma only accounts for 5% of pediatric cancer, but it accounts for 15% of cancer deaths. So it's a very important tumor in children, and it has a highly heterogeneous behavior. This ranges from spontaneous regression to rapid progression and death. So in some children, the tumors just disappear on their own, and in other children, in spite of highly intensive multimodal chemotherapy, the child might die. So part of my role in Crumlin was to do genetic diagnosis on the tumors to get prognostic information that could be used to make therapy decisions on treatment. So if the child had a tumor that was going to spontaneously regress, you don't want to give them tons and tons of highly toxic chemicals to try to make the tumor disappear. So genetic diagnosis of this particular tumor is very important. And one of the diagnostic genes is the oncogenic transcription factor MYC-N. Children with tumors with MYC-N amplification have only about a 50% chance of survival. Even today, only a 50% chance of survival. By MYC-N amplification, I mean that what happens in the tumors is that rather than having 2 copies of this gene, one on each chromosomal homologue, you have maybe 200 copies of the gene. So the gene has been duplicated many, 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 many times, and you use a technique as seen here called fluorescence in situ hybridization. The pink staining is MYC-N, and you can see there's a couple of hundred copies of MYC-N in the nuclei of these cells. And on the right, you see a Kaplan-Meier graph showing that in the blue, children with MYC-N amplified tumors have very, very poor survival. And this is highly, highly statistically significant. You see in this cohort of tumors that we developed, the p-value is to e to the minus 17. So as I said, this was a major career shift for me going into diagnostic work. And during this period, this 9-year period that I spent in Crumlin, in addition to doing diagnoses, we were also able to do translational research on neuroblastoma as well. So one of the little things that I'll say to you in terms of career advice is don't be afraid to alter career pathways, but you should do so with the understanding that the change could be permanent. Now, for me, I was able to move in and out of different careers with little difficulty, but do be aware that a career change can be very beneficial. I found it very beneficial, and it can also lead to, but it could also be permanent. You may not be able to go back and forth. So I wanted to work more on neuroblastoma in terms of translational medicine in basic research of the tumor. as opposed to doing routine diagnostic work. So the University of Texas Health Science Center in San Antonio opened a brand new Children's Cancer Research Institute that was very well funded. Actually, it was funded by money from the tobacco industry, of all places, because the states all received court settlements from the tobacco industry. And Texas decided to use their money to set up an institute to study children's cancer. And one of the first major things that I did on this tumor was to use a technique called comparative genomic hybridization, CGH, on microarrays that were produced by a company called Nimblegen. And basically a microarray, these microarrays are quite remarkable devices because they're the size of a microscope. Okay, there's a message that keeps popping up, start my video. I don't know what that's about, but I'm just going to close it. But these microarrays are the size of a microscope slide, and this company was able to synthesize in situ 1 million different oligonucleotides on the slide, on the piece of glass, the size of a microscope slide, that represent different regions of the human genome. And by doing this and by doing comparative genomic hybridization, I don't really have time to explain the technique, but basically it's a technique that shows loss or gain of genetic material in the tumors. So we were able to map very subtle regions of genetic loss in these tumors, as you can see right here. This is like some of the output from the microarrays. So for example, we could show there were microdeletions in the protein tyrosine phosphotransferase receptor gene, PTPRD, and we were able to document that this was a bona fide tumor suppressor gene in neuroblastoma. So there we go. In addition to being able to use these arrays to study the gain and loss of DNA sequences in tumors, you could also use them to study microRNA expression profiling. You could study the expression of the total transcriptome of messenger RNA sequences, looking at levels of expression. You could use them to map sites of DNA methylation in the promoter regions of all the human genes. And you could also look and identify MYC-N binding sites. So MYC-N is that transcription factor that's amplified in neuroblastoma. So we were interested in identifying which are the genes that are being regulated by MYC-N binding. And so this was the first time that anybody had taken a set of 200 or plus tumors of any type of cancer and to look at microRNA expression profiling, mRNA profiling, DNA copy number changes, DNA methylation changes at every promoter region for every human gene, and whether or not MYC-N was binding to genes or to microRNAs, because we did in fact find that MYC-N regulates a large number of microRNA. So we then focused our studies primarily on the microRNAs, and we found that there were many microRNAs whose expression profiles could be linked to survival in the children with neuroblastoma tumors. So for example, as you see with these Kaplan-Meier graphs, miR-92 is upregulated. It's expressed at high levels in tumors where the children have very poor survival. Conversely, miR-204 is expressed at very low levels in tumors from children that have poor survival. So these microRNA turned out to be really extremely important biomarkers for predicting survival and will eventually be used to guide therapy. in addition to doing the profiling, we did a huge number of functional studies on many microRNAs. So for example, we identified that miR34A is a tumor suppressive microRNA in neuroblastoma. This was the first paper. We published the first paper showing that miR34A is involved in any form of cancer. And it turned out to be a major tumor suppressor. Since we published our first paper on miR34A in 2007, there's been more than 1,200 publications on miR34A and cancer, and we published the first paper, and our paper has been cited quite extensively. And the reason miR34A is a tumor suppressor is because it is multi-targeting. It targets dozens of oncogenes, like it targets the MYC-N oncogene itself. So there's no point in a neuroblastoma tumor of having amplification and overexpression of MYC-N, for example, if you have a microRNA that targets it and knocks down the expression of the protein. So miR34A is multi-targeting. It targets huge numbers of genes that need to be upregulated in bad forms of cancer, particularly in neuroblastoma. Following on from miR34A, we did functional studies in vitro and in vivo of a lot of different microRNAs. So, for example, we showed what their targets were. So, and we also showed how they affected different aspects of cellular phenotype. So miR34A, for example, regulates apoptosis. miR10A and 10B regulate differentiation. If you overexpress these miR10A and 10B, it causes neuroblastoma cells to differentiate. Some microRNAs affect the ability of cells to invade other tissues and to migrate and to metastasize to other sites in the body. There are microRNAs like miR204 that impact on drug sensitivity and affect how chemotherapy affects tumors. I mentioned earlier that we could also use the microarrays to assess how DNA methylation at promoter regions of genes, looking for micro, and we did this primarily for microRNA. We looked at the promoter regions of about 450 microRNA, the genes that encode for microRNA, and identified which of these microRNA were regulated by DNA hypermethylation. So here's a list seen here where we correlated DNA methylation at their promoter regions with expression. And we showed that microRNA that had hypermethylated promoter regions had much lower levels of expression than microRNA that did not have methylation. We developed and showed that there is actually a large set of epigenetically silenced microRNAs that redundantly target a large network of clinically relevant genes, more than 1,000 genes that are upregulated in neuroblastoma are targeted redundantly by microRNAs that are epigenetically silenced because they're tumor suppressive microRNAs. For example, one gene that is Lin28B, this is targeted by a set of about 10 microRNAs that are epigenetically silenced. Lin28B needs to be upregulated in neuroblastoma. And we actually had a major international collaboration, and the results were published in Nature Genetics a few years ago, where we showed that Lin28B is actually a bona fide oncogene in neuroblastoma, because if you artificially upregulate Lin28B in mice during mouse embryonic development, these mice develop neuroblastoma tumors. We had to develop our own bioinformatics software that we published in Nucleic Acids Research for analyzing all of these different types of data and integrating them together. And then finally, we explored the possibility that miR34A and other microRNAs can act not only as biomarkers, but as potential therapeutics. So what we did was we encased miR34A in nanoparticles, and we injected them into mice that had neuroblastoma tumors, as seen here. And we showed that miR34A encapsulated nanoparticles could slow down tumor growth in the mice, as seen here. Other groups, particularly in the US, have actually done clinical trials with, and I'm not part of this, but they did clinical trials with miR34A that was encapsulated in liposomes and showed that it had some benefit to patients, but the clinical trials had to be stopped because of severe immune mediated responses, adverse responses. So there is potential for microRNA replacement therapy to be used in cancer. We did a lot of work on studying the role of microRNA in other types of disorders. So for example, we collaborated with people at other individuals at RCSI that showed the role of microRNA in epilepsy and in autoimmune disorders like SLE. As I mentioned, I then changed careers and became director of research. I won't go into that to any great extent. I did have a very large research group. This is them there. This was a very happy time. Many international collaborators that I won't mention or go through in any detail. And then just to leave you with a little bit of career advice. I mean, it's very important to publish your papers in highly ranked journals. Think about quality over quantity. Be resilient and be prepared for failures. I've had 5, 6, 7, 8 failures for every success. Be altruistic and generous to your students, your colleagues, and your institute, because you'll be rewarded for that kind of behavior. And don't be shy. Seek out advice, particularly from senior colleagues. So I will end my talk now and answer any questions that might arise. Thank you. I guess I will unshare the screen.
Yeah, I think your camera turned off in the middle of your talk there. You should be able to enable it again now.
Okay.
Oh yeah, perfect.
Were you able to see the slides?
Yeah, we saw them.
Yeah, okay.
So I'd like to thank Professor Stallings for his fine speech. And I hope that a lot of you were able to get some stuff out of it. So I just have 2 questions I'd like to ask you. So firstly, what do you think has changed the most in the process of research over the course of your career and your career changes?
What has changed the most? I think, oddly, what has changed the most has nothing to do with science itself, or maybe it does. I think what has really changed is that there's a huge population that no longer believe in science. And this is a lay population. And so I think we're going to have to rethink how we go about trying to educate or re-educate people. There's so many conspiracy theorists, and you look at the response to the pandemic in the US and also in European countries, like there's been rioting in the Netherlands, and nobody seems to understand the seriousness of this pandemic. And I think that has been this didn't exist, this kind of attitude didn't exist when I began my career.
Okay, that's great, because we actually have Ruth Freeman. She talked earlier, and she's the director for science in SFI Ireland. So it's interesting to see that media connection and trying to get the word out there.
Yeah, very much so. And the other thing that may have changed is PPI, patient public engagement. And that is just ever so important. And it relates to what I said just a second ago about, the disbelief in science now that is rampant.
Great, thank you very much. So I'd like to ask another question from John Matthews. Is there a subpopulation of cancer stem cells at the core of neuroblastomas? And if so, has there been any treatment options developed against this subpopulation of cancer stem cells at the core of neuroblastomas?
Well, that's actually the core of the problem is the cancer stem cells. And yes, it is considered that there is a core of stem cells in neuroblastoma. They are very, very difficult to treat, possibly because some of the stem cells are hiding in places where chemotherapy can't penetrate. The difficulty in any type of therapy now in cancer is really getting at these stem cells because they're the ones that will continue to repopulate metastatic sites. There is no specific treatment to date for the stem cells. I mean, in some children, in 50% of children, for example, with MIC-N, they do survive the cancer. So presumably the cancer stem cells have been eradicated. You can have children that have where the cancer has disappeared for 2 or 3 years, and then suddenly it comes back, undoubtedly because of a stem cell population.
Okay, so we just have 2 minutes if you'd like to answer a very, very short question for myself, actually.
Sure.
So you were talking about this cancer in children and how it's so enigmatic that it might relapse or it might not, and such a high mortality rate, respectively to other cancers. So is that what motivated you to pursue this career path?
Well, that was part of it because, there's no point in working on a cancer well, there is plenty of points of working on cancer to find alternative therapies that are less toxic. But certainly I wanted to work on a cancer that had a huge impact on children. And this was a particularly nasty form of cancer. And it occurs mostly in very young children before the age of 5. And it affects particularly badly, it affects kids when they're at toddler age. And it's particularly devastating to parents because you think you have this, you have this fantastic, bright, bubbly little 2-year-old, and suddenly they become very, very ill.
Perfect.
So it's a good motivation for going to work.
Yeah. That's wonderful. Thank you very much. So I'd like to close this talk now. Again, thank you very much, Professor Ray Stallings. I'd just like to let you all know that the next talk will be from Ali Rose Fisk. And I'd like to wrap this up. Thank you again.
Thank you.
Thank you, Ray. Bye.
Bye.